Hello students, this is Faizan Mirza. We are discussing gene technology and its application in medicine. The common most vectors which are used for genetic engineering, these include prokaryotes, eukaryotes, viruses, and liposomes. We studied how prokaryote, the bacteria E. coli, was used in the production of human insulin. So the bacteria, eukaryotes, for example, the eukaryotic tissue which is commonly used could be the tissue taken from eukaryotic cells and grown as tissue culture in lab. And viruses such as retroviruses, they can be used for delivering gene. Lentivirus can also be used for the same purpose. Adeno associated virus, AAV, can also be used for the same purpose. Liposomes, these are artificial vesicles of phospholipids. They can also be used as vectors. And you can see that the vectors which have been used so far, for example, human factor VIII can be produced using eukaryotic vector, modified hamster cells. This is an example, and it treats hemophilia. Human antithrombin is it reduces blood clotting. Goat cells are used here. Human alpha antitrypsin, this is from sheep. It is used. The sheep cells are used here. So this helps in management of emphysema. Ovarian kidney cells are preferred and they are cultured in fermenters. What is the basic premise of gene technology in medicine and in the field? How is it applied? So the basic principle is to deliver a working copy of the gene into the cell which initially was lacking it. So, through this technology, the gene is incorporated into the cell, and this makes use of a vector. And so the host cell, it uptakes the working gene, and when the working gene is incorporated into the genome of that individual or the host cell, the gene ends up getting expressed. And when the gene gets expressed in the host cell, the polypeptide of that particular trait gets shown in the phenotype. So it is used. How is it applied? First of all, the genetic screening is performed, collection of sample, just amplifying it and analyzing it, PCR, electrophoresis, microarray, and then going for gene therapy. Gene therapy means how the affected cell can be given the actual gene using a vector, so the gene becomes a part of that affected tissue and the genetic disease gets cured. So how genetic screening is done. So let's start our discussion of genetic screening. The analysis for the presence of a certain gene in the person is done, and the person's DNA is checked for the presence or the absence of a certain allele. Adults can be screened for genetic disorders such as thalassemia or sickle cell anemia or Huntington, and even for cystic fibrosis. Especially these are very grave disorders and cancer predisposition, yes. And even not only adults, the fetus can also be screened for possible genetic anomalies, and especially if the parents are the carriers of that same recessive trait or the dominant trait, for instance. And if that's the case, then therapeutic abortion is suggested to the family. What is therapeutic abortion? The fetus with the genetic anomaly or abnormality can be aborted for on medical grounds, because the fetus, if it develops into a baby and is delivered, it would have that genetic deformity which cannot be treated that easily. So how sample is collected for testing for the possible genetic defects in a fetus? There are two techniques which can be used: amniocentesis and chorionic villus sampling. So what is amniocentesis? So, what is amniocentesis? In amniocentesis, the fluid, which is amniotic fluid, it's collected as a sample from around the fetus. This is done at 15 to 16 week of pregnancy. This technique is carried out using ultrasound guided hypodermic needle, which is inserted into the abdomen of the female, and the needle is directed away from the fetus umbilical cord and the placenta, so these structures are not damaged, and only the amniotic fluid is collected for sampling. The other technique is chorionic villus sampling. In this technique, a small part of placenta, which is called as the chorion, this is removed, and this is scrapped slightly, and this is done at 10 to 13 week of pregnancy, and this is done using an ultrasound guided catheter which is inserted through the vagina of the female, and the sample is drawn from that region, from the chorion of the placenta. So this is one technique. These are the two techniques which are deployed. The samples which are collected, they can be analyzed initially for a karyotype, and in the karyotype you can see for a chromosomal anomaly, and if not for a, if the karyotype is normal, then the gene testing or the testing of for certain alleles can be carried out. So here you can see the process of prenatal testing, and this is called as a prenatal testing. Prenatal mean that the testing which is done before the birth of an offspring. These two diagrams here, this shows to you the chorionic villus sampling. The catheter is inserted through the vagina of the female, and you can see that this is the region, the placenta. This part is scrapped. A chorion is removed, and the catheter it's drawn back. So this is the fetus. You can see that the fetus is not really it's not really you can say damaged, or it's not really harmed in this way. And since this is done a bit earlier, 11 to 13 weeks, so you can see that the fetus is a bit not that large. And this is ultrasound guide, so it constantly tells the position of the fetus in the womb of the mother, so when this test is being performed. In amniocentesis, this is done a bit later, so the fetus is a bit larger by now, and again you have to use an ultrasound guided needle. So the needle is hypodermic needle is inserted. You can just see that this needle is not the kind of a small syringe which is used for blood drawing. This is a bit larger, because you are drawing a different sample, a different fluid altogether. So this is done by this technique, and this you can say the fetus is a bit large here. So anyhow, the amniotic fluid will have the cells of the fetus, because the fetus cells which are getting sloughed off from its body will be present in the sample of the amniotic fluid. So these fetal cells can be collected, and they can be studied for genetic screening, and the same can be done with chorion as well. So that's it from my side. Thank you so much.